All right, everybody. How are we doing today? Can everybody hear me? Awesome. It is the final night of See and Learn 2021. So we're going to get started in just a moment. Please grab your drinks, find your seats, say hi to the person next to you, buy a raffle ticket, and other than that, we'll get started in just a moment.
Good evening, everyone. Welcome to... Welcome, everyone. Thank you for coming. And this is the final night of See and Learn 2021. I'm not tall enough. Thank you. I'm going to do a quick recap because we want to celebrate everything that was done this month. Before I do that, though, I want to give a great thanks. I want to hear a big shout out for Emily. Stand up. And JJ. They have worked absolutely tirelessly the entire month, every single day. And knock on wood, we still have some school stuff tomorrow, but we didn't have any glitches. So they did a fantastic job. So thank you very much. So this was a fantastic month. Some people are here for maybe only their first or second time. Some people in the crowd have been here almost every night. But this is going to be just a quick 10 minutes to run you through what a great month it was so we can all reminisce. We started on October 1, as we always do. This is with Dr. Edie Witter. And she is an expert in bioluminescence, deep water sea creatures. And because of that, because of her bioluminescence knowledge, she was invited on a deep water sub, and she was one of the first people to get video of a giant squid. And she took that information to our high school kids, and yeah, totally awesome woman. Next up was Erica Moulton, and she works with underwater remote operated vehicles. So it was we started off the month with deep sea exploration. And understanding that, but what was really cool is she showed our school kids, it's a SABA comprehensive high school children, as well as adults, and showed them how to make their own ROV, starting with a coat hanger. And look how fun that was. Then we went up to the Eco Lodge, and these were special evenings in the rainforest, so TED Talk in a rainforest. What about that on SABA? And this was Elska and Reynold, and they are earthquake and volcano experts, and they taught us all about how to monitor our own quiet, active volcano. They took school children and adults to the monitoring station in St. John and the equipment in St. John where you can see what's going on. And then here's some stuff everybody really enjoyed. So some people are afraid of scorpions, spiders, and snakes, but how about the kids up here? No, it was cool. And we learned how to find them outside. So they went to the classroom and saw and showed kids how cool all these things are, up close and personal. But then we went on our first night hike, and we had at least 40 people And we went on a real hike and found all kinds of cool stuff. Next up was Tadzio. And Tadzio grew up in St. Martin, but he now lives in Bonaire. He's the head of Dutch Caribbean Nature Alliance. And they did an expedition in July on the Saba Bank, specifically capturing, short term, not killing, and looking at pregnant tiger sharks as well as other sharks. We couldn't duplicate that, but he gave us a fantastic presentation and took people out on a shark dive, so that was just another highlight. It was Shark Weekend, and then next up was Mitch Ryder. And Mitch studies acoustic and satellite telemetry and how we track sharks in different ways. So he took the after-school program to the Cove Bay, taught them all about how they do shark workups and how they monitor them throughout the Caribbean, and everybody had a great time. Then we had Michael Middlebrooks. Oh my God, you talk about enthusiasm. He's not a slug, but he taught us all the cool stuff about slugs, and we did that at the Hell's Gate Community Center, where the Saba Comprehensive School Hospitality Program hosted the evening, serving food and drinks. It was really a special evening, and the take-home was that slugs are snails minus the shell. But he taught us about solar-powered slugs. And he started with school children down at the Cove Bay, which is a beautiful, picturesque place. They took some things out of the water, got to examine those, and really gained new knowledge. And some people were even lucky enough to dive with them. And Saba has all different types of slugs, nudibranchs, so cool stuff. Then we go to orchids. Some people are like, oh, plants? Well, you talk about enthusiasm. Our orchid expert, super duper knowledgeable. It was a great presentation, but even better than the presentation was there was all kinds of field projects with kids and adults. So he is part of the research of orchids on Saba. There's about 3 different guys who have done it. And believe it or not, just in that part of the island, there's over 800 orchids that they keep track of. And this isn't the easiest place to hike, as JJ and I found out. And these are mapped, identified, GPS, tagged, and they're monitoring their health and, sorry, help me out, JJ, how much they grow, size. But yeah, and there's also orchids just that are monitored right on the Dancing Place Trail. That's where the school children went. A new appreciation for, again, a telltale species. Then we go to Mark Butler. He taught us the Lobsterina dance. How many people were here for that? Yay! Okay, so that was fun. He also did a night dive. But the other cool part about See and Learn is not only does it give our children role models and learning that science is fun, because all these experts are truly educators, they have ways of sinking in information that kids are going to remember for life. I quizzed a 5-year-old the other day, and I couldn't believe what she retained from that school project. Here, he had them make pinwheels. Well, why would a lobster expert make pinwheels? Well, because lobsters, when they travel across the ocean floor, they actually use drafting, like race cars do, like some other animals do. So he had to make pinwheels and then walk outside and count how many rotations the pinwheel was doing. Then they did it in a line. And then they realized how lobsters can move faster and use less energy. And I'll bet you those kids will remember that for quite some time. But again, not only are they educators, they're entertainers. So look at the joy he brought to the faces at the Life Center, because he played a game and they did a critter bingo. So this is just another example of the cool things that happened during the month of October with See and Learn. Then we have Kim Bassos Hull. And this is another great example that See and Learn addresses from K to gray, as we say. So from kindergarten through the Elderly Life Center. And she's a ray expert. So stingrays, eagle rays, manta rays. But she also is knowledgeable about dolphins. So she studies things that jump out of the water. So when they learned that she was also an expert with dolphins, she also worked with the after-school program down at the Fort Bay. Obviously, they had a great time. But then she went on her little expedition. So courtesy of the Saba Conservation Foundation, who utilized their boat, they went out to deploy a bruv. Who knows what a bruv is? A bruv is a baited, help me out here, come on, a baited remote underwater vehicle, okay? And here you can see it, you put a camera on it, they put it overboard, they find the right substrate, and then they leave it. So this is another way scientists work to find out how animals behave when we're not there. So if you're down there diving in a lot of bubbles, it's a whole difference than if a camera's there just by itself. And then there was a dive on that to be in action, and eventually they got to look at that footage. Next up was our very own. So what is this? If you're down at the harbor, you're like, okay, here's a container. If you open up the container, you might find this Dutch guy. And he does really cool stuff in there. It is Saba's own laboratory. And this is Elwin. Elwin doing the Moe Reef project, which is diadema or urchin restoration, which we did a dive today. We were happy to say we actually saw a whole lot of urchins in a place we didn't expect to, so positive things. But if you haven't done this, many people were exposed to it, but they are willing. Some of the team is here tonight. Hello. But they are all about outreach and sharing this knowledge of what they're doing to make a difference on our reefs, both so you can go to the laboratory and they'll explain it to you, but they also worked with our school children and adults looking at the larval stage, going to see actual size urchins and how we eventually put them out on the artificial reefs they've created and gauging their artificial reefs compared to other methods. So this is a project being done on Saba and Stacia, and they are seeing success. Next up was Lara. Lara's here tonight. Yeah. So she did a mini presentation the other night about her work with tropic birds. And tropic birds are found in the region you can see across the kind of equatorial area of the world. But Saba and Stacia are very important nesting grounds, feeding grounds for these beautiful birds. And there's studies being done on Saba and Stacia, and we represent, what's the magic number? Almost 25% of the world's populations are dependent on Saba and Stacia. So thank you, Lara. But Saba, or See and Learn, is not just about hiking and diving, and we also sometimes address the real unknown, space. So this was our wonderfully enthusiastic astrophysicist. And this is, again, a fabulous educator who can take something like massive black holes and go to kindergartners and first graders and really teach them about planets, stars, the sun, and what's bigger, heavier, denser. And it's amazing what those kids absorb from those couple days. See, you were there? Okay. I saw Hugo there. And then we did a night walk the other night, 46 of us, 47, we don't know, might have left one behind. It was dark. The sky wasn't super cooperative, but it was still pretty interesting. And he brought his microscope, and this just gives you an idea. He was using a laser to teach us all about the constellations, where we can see them on Saba. So cool stuff. Last night, so as we're almost near the end, Carolyn Caparuso, I think I just saw her walk in. Yeah, she talks trash. And as she said, maybe not as sexy and entertaining as some of our sea creatures, but what See and Learn also reminds us all is, in order to appreciate all the cool stuff we see in nature, we have to protect it. And it was a little depressing learning how poorly the world is doing managing our garbage. But she brought solutions. We went diving this afternoon, but this is samples from Bonaire. And again, it doesn't mean it's Bonaire's fault, but this is actually from this afternoon. an abandoned fish trap that was there, but it was cut open, so there weren't any animals being caught in there. We only found 20 pieces of trash in a pretty big area, and there was 7 of us diving, but it still shows that, yes, marine litter is out there. But Carolyn and Alex are doing a great job in Bonaire. She works with World Wildlife Fund and the United Nations, working on getting a broader platform to address this issue. And this is what they also do, is they take ocean trash and turn it into usable things. So if you did not get your field project gift, they are still available today and tomorrow. And these are the sort of things you could do with it. So turning trash into positive solutions, plant a tree. So tonight we have our final speaker that we're going to segue to. And these are images from his school project the other day. So he took kids out of the classroom. So we like to take kids out of the classroom, get them with hands-on things to show them methodology of how you do certain things. So Andy's going to tell us more tonight. One of the things he works on is how you calculate conch population using quadrats. So he can explain the process that he did with these kids and why it matters. And this is just a little, who's that? Young scientist. This is just a graphic of the type of stuff he's going to talk about tonight and why larval dispersion matters to all of us. And that's what I have to say tonight. I think we should all feel really good. We had a great month, and we're going to look for you in 2022.
All right, give me just a second. She's pretty short. All right, now, like Lynn was saying, we're going to have a great year next year as well. One of the best ways that you can actually keep up with us is our Facebook page, See and Learn. So that'll keep you connected with us all throughout the year. And then we will be back for See and Learn in 2022. Same place, same month, same purpose. So definitely come out. Thank you guys so much for all your support. And we have a lot of people that support See and Learn. It's a lot of things that it takes to make this happen every year. So as always, the live streaming is sponsored by Carib Trans, so big round of applause for Carib Trans. You guys can watch this presentation or any other of our presentations online on the Facebook or on YouTube, all right? So I did not just say the Facebook, by the way. We're going to cut that from the live video. I swear I'm not that old. Yeah, so definitely check those out. Now, our speaker tonight is being hosted by Queen's Garden Resort, so big round of applause for Queen's Garden Resort. We've had a lot of different hotels and villas around the community that have helped us out this month, so we are very appreciative of them. Now, of course, none of this would be possible, even in the first place, without the Prince Bernhard Culture Funds or public entity SEBA. They are huge in this, so yeah, a big round of applause for all of them as they help us out every year. And to pretty much all of the businesses that are on SEBA. It takes a lot of things to make this work out, so big round of applause to all the businesses. And I want to give one big round of applause to you guys as well. We wouldn't be here if it weren't for everybody coming out and making this possible. So give yourselves a hand. All right, now I'm sure we've hammered those raffle tickets into your brain very well. So real quick, a little what you could be winning tonight as we do the live drawing right after the presentation tonight. There is an indigo pareo made by Anna Keen. Very, very cool. We actually have it here tonight, so if you win it, you can take it home with you. That's my Vanna White over there. Now, we have some handcrafted jewelry, some homemade Saman Rock Living beauty products. We have a signed copy of Edie Witter's brand new book, Below the Edge of Darkness. We've got a guided hike from the SEBA Conservation Foundation. We have got a class with Jo Bean herself to do some glass making, and she's giving away 2 sculptures that she has professionally made as well. So that is awesome. It's all in one big bundle. We have got several cottages that are giving away 3-night stays. So that is crazy value. It's at least like $300 or $350 a night for each of these. So definitely that is awesome for when you guys come back to see and learn next year in 2022. We have got a handcrafted knife from John. It will be custom made. So definitely check that out. And an 8-day, 7-night liveaboard on the Caribbean Explorer. All right, fully paid for, all your dives, all your food. I'd go on there just for the food, honestly. But yeah, it's a crazy value that you're getting for these. So I hope that you didn't miss out on buying raffle tickets, because we'll be doing the drawing right after the presentation tonight. But for tonight, we have Andrew Cow. All right, so with a name like Andy Cow, how can you not be a nature lover, right? So Andy is a great guy. He was actually doubles as the Flash, believe it or not. He was able to make it here on very, very short notice to help us out when our plans changed a little bit for the final night. So without him, this wouldn't be possible tonight. So big round of applause real quick for Andy. Thank you for coming out. Now, Andy, he has a PhD in marine biology from the University of Miami and has been lucky enough to work on projects funded by the National Science Foundation, NOAA, and the U.S. Fish and Wildlife Services. He has studied and published scientific papers on a variety of species, including corals, marine fish, sturgeons, sharks, spiny lobsters, and the majestic queen conch. All right, so let's give a warm welcome to the conservation king himself, Andy.
Awesome. I'm starting a timer so that I am on point tonight, because we're already running behind, no doubt. So before I talk to you about my favorite thing in the whole world, which is larval dispersal, the foundation of every marine ecosystem, awesome. First, you're going to hear, because you're a captive audience, about 2 minutes about who I am and why I'm here. And then you can use that to judge whether or not you want to just drink your beer and zone off or actually pay attention. Okay? So my last name is Cal, like the animal Moo. I was born in Washington, D.C., this house, this dog. And I've always been really into nature. Check out the swag on my feet. Yeah, awesome little froggy boots. Unfortunately, they don't fit anymore. So every single picture of me when I was a kiddo, I am holding or harassing marine life. I mean, life. Animals. And that's a trend that continues to this day, but not in Saba. I got corrected and told that I couldn't touch anything here, and I didn't do it. Snakes. They've played a really important role in my life. Everyone loves snakes, right? Yeah, absolutely. So these really got me into science, especially baby girl, rest in peace after 19 years. Because I realized that a lot of people have different thoughts about these animals, and you can talk to them, take care of these animals, and then dispel a few myths. And I worked at a couple of nature centers in high school, and then throughout college, I worked at exotic animal rescues with a bunch of scary things. Because I wanted to be a vet. Although I'm a super positive person, and I worked at a vet office for 2 weeks, and that's all it took. Sad dying animals, sad unhappy people, that was not the life for me. So back to the drawing board and back to nature. I was lucky enough to get a position studying birds, white-throated sparrows, in basically Canada, where it snowed in June. Sounds pretty disgusting for here, but it was cool. And along the way, I also had a couple more transformative experiences. It was awesome to study birds at dawn in a swamp. It was also cool to go to Australia, not because of the fuzzies, but because of the Great Barrier Reef. And this is the first place that I ever dove in my life. And once you descend beneath the waves there, how could you not love this? There's all this color, texture, noise, everything you could possibly want for a good time. And I was immediately jealous. Like, these kids are so lucky they get to grow up next to the ocean as opposed to in a landlocked area where I was going to college. So I had to settle and build a fish tank. I somehow convinced distributors from around the Indo-Pacific to send me rock, and then I did an undergrad thesis looking at the little animals that popped up on that rock using electron microscopy. It was cool. After school, I didn't really know what to do, so I worked for the Fish and Wildlife Service. These are ponies, and I hate them. They will ruin any picnic, and they will attack and bite. Praying mantises, on the other hand, super cool, useful, beautiful, and I had a second job working for the local Department of Natural Resources, looking at how different insect populations around are interlinked, which sort of sent me on my way down to Miami, where I did a PhD in marine biology. And I just really want to point out right here, if you ever go to Miami, go to the wet lab, okay? This is a bar entirely run by grad students, very reasonable prices, and you can see the sun rise from this side and set from this side. So it's a really scenic, awesome place to go for a cheap drink. That was my grad school experience. I also tend to write haikus during talks because they're fun. So I'm gonna break up all the different sections with a little poetry. All right, so here's your first one. Born naturalist, chose ocean science career, loves aquariums. Work for one. And invertebrates. Look at that cute little thing. Okay, so on to larval dispersal. So I think this is one of those unknown and extreme processes. So when you think about dispersal, you're often thinking, if you're me, about animals that spend a substantial amount of their time and energy dispersing, especially as adults, right? So like the great migrations that a lot of birds do, or perhaps the wildebeest chasing the rains down in Africa, right? There's a whole missing group. Things like plants that are lazy. They're planted, they're really rooted down there and they're not moving too much, but their seeds still are, either through animals or by harnessing the physical environment and hitching a ride. Lots of animals take this approach. So like spiders, for example, ballooning off. But that's not to say that these are passive dispersing animals, because they still control a huge amount of their destiny. So a plant could release seeds during a specific time of year where they know the winds are going to be more substantial. And I can guarantee that if a spider jumped off of a blade of blade of grass versus climbing a redwood, it's going to be a completely different trajectory that it's on. And that's more similar to what's happening in the ocean. So all of those fish, all of those invertebrates, peanut worms to crabs and lobster and fish, they all are harnessing the power of the ocean to move from place to place. They form this incredible planktonic assemblage that without the plankton, you get no baby fish. No baby fish, bye-bye fishies and reef. And that's known as the complex life cycle. It's also known as a bipartite life cycle because you have two completely different stages that the same organism has, right? So this little thing and this big old, I guess, post-larvae look completely different. And they also fill different roles. So a shark, or I guess more aptly, a grouper or something might eat this when it's an adult. But these larvae might be feeding on grouper larvae while they're out in the plankton. So it's a super cool, interconnected, and hard to study thing. It basically encompasses two processes and two different sets of habitat. And we're going to talk about both of them. Dispersal, so that's out in the open ocean, moving from place to place. And then recruitment, which is that return journey from the open ocean to find a forever home on a nice island paradise like Saba. And we're going to talk, there basically it's a chicken or the egg thing, stupid joke. We're going to start with recruitment, right? So finding a benthic home from the open pelagic, key life transition, and look how cute, all right? So it's awesome to work with baby animals because they're adorable. So I want you to put yourself in your mind right now out into the position that a larval animal faces when they're trying to make that transition back to a coral reef. So if you're at the surface out in the ocean, you have this ever-moving matrix of wind and waves and no signal. If you start descending a little bit more beneath the water, you might get lucky and have some sort of cool celestial cue overhead. And that is the environment, no map, no features, that these animals have to navigate through in order to find their way to a coral reef. And they're also really strong swimmers. You don't want to swim strong without a purpose. We don't understand a lot about what's guiding these animals. So I'm going to give you one of my favorite animals to work with here, the spiny lobster. They get spawned off the coast of a lot of islands, and they make this incredible journey where they feed, get bigger, they go up and down every single day. They're really flat, so they slice through the water column until finally the need hits, and they transform from this alien-looking phyllosome into a post-larvae. And the phyllosome comes from the Latin root of leaf, because they are completely flat, and they look like weird spiders while they're riding jellyfish. And then if they're a leaf, then the post-larvae is certainly a rocket, because their whole purpose is to jet back from the open ocean to the coast. How do they do that? That was a question I asked. Oh, there's noise for the overhead. So they're also really adorable. They're extremely invisible in the water. So if you take them out and put them on like your fingers, it looks cool. Also the last good use of an overhead projector, because you can't see them, but you can put them in a petri dish on top of it and watch them swim. How do you track something like that? So this is an animal that's so small that it can perch comfortably on my finger. You can't put a satellite tag on it. And even the smallest tags that we can use to track birds are about the size and weight of a penny or a dime now, probably. And obviously a penny or a dime is way larger than these animals. We haven't yet developed the technology. So maybe like 20 years from now, these kids are gonna know what's up. I don't. I had to improvise. Grad students use glue, PVC, and sheer luck. So I started leashing lobsters. So I would catch them during the night, I would put a little leash on them, and then I would take them out into a behavioral arena during the day and the night to see where they were swimming. I know where the arena is, I know where the wind is going, I know what sort of tidal cycle it is, and you can watch them swim. They swim with their antennas straight out in front of them, furiously beating their little pleopods, like the cleaner shrimp in Finding Nemo. They swam. Also, I built an infrared lighting array so we could watch them swimming at night. You don't want to miss half of the process. Yep, so they can't see it at all. And also, we only lost one to predation the entire time, and it was another lobster that somehow appeared. Yeah, I thought I was delusional at 3 a.m, but there was a lobster that was on top of the trap when we pulled it up. It's cool. As Mark told you, they are very good chemosensors, and maybe it smelled a snack in a competitor. What did we find? So we just built a simple model to build off of our findings. So if you put a little larvae and release it in the current speed that we saw during the experiment, it ends up there. However, if you use the different behaviors that we observed with a change in ebb tide, so that's when the scent of near shore is offshore. So these animals can smell, hey, land is nearby. It's time to do something. And they had a completely different behavior, which turned out to be very advantageous for them finding a home. However, odor in the ocean is not a directional cue. It goes spread out by turbulence instead. So this is more of a flavor that says, okay, I'm nearby, time to activate the hidden senses. And what we found that the lobsters were doing was orienting relative to the wind. So only the animals at the surface were able to perceive where to go. And in Miami, there's a persistent sea breeze that points them towards the coast. So these animals are tasting the delightful flavors of offshore Miami and then surfing towards the coast. And that's happening all over the world. Super cool. There's a haiku for you. And those are my fingers. Okay. What other cues are available? So sound. The reefs are super noisy. Anytime you go snorkeling or diving on a reef, you're gonna hear it. It's all these cracks and cringles, and then even the waves breaking or the cars going by. All of this makes this incredible soundscape that these animals can home into, and it's directional. We already talked about odor, smelling some exhaust, but there's other cues, the wind, the waves, also The sun. Some larval fish have been shown to have a sun compass, so they can orient where they're going relative to celestial cues. And also others, magnetic. So the little leptocephali, which are the final stage of eels, bonefish, and tarpon, are able to have a magnetic sense to find their way. Oriented swimming is good swimming. You don't want to just swim in a circle. You want to know where you're going. Cool. So now we're going to have a short video from my lab mate during grad school that talks about sound for like a minute.
Every year, billions of baby fish are born into our
ripped.
oceans to face an uncertain fate. After traveling for several weeks as pelagic larvae, these fish have to find the proverbial needle in the haystack, a coral reef. But how do they find the reef? One potential navigational cue that they may use is the reef soundscape, which sounds like this. For my dissertation research, I am recording the sounds of a coral reef in Florida to understand how it changes over time and to identify the biological and abiotic components. Then I play back these recordings to fish larvae in the field using a unique behavioral chamber coupled with a pair of underwater speakers. This allows me to see whether these fish larvae can orient towards reef sounds. Coral reefs comprise less than 1% of the ocean, but they are one of the most important areas on the planet, both ecologically and economically. Due to human impacts like overfishing and climate change, they're also one of the most threatened marine habitats. We need to discover how fish larvae find their way home, because the replenishment of reef fish populations depends upon the success of this next generation. Understanding the navigational cues used by larval fish will help us to protect coral reefs for now and the future.
And sound pollution is a problem above the waves and below the waves. So lots of different animals release millions of eggs, which leads to one question. Why aren't we overrun with fish? Well, the reason is super grim. The average larval fish is in fact dead. Sad. So along the way, normally when we study them, we'll use something like a light trap to collect them. So they are phototactic. They will come towards the light, and we can catch them on the way. Here's an example video, which would have been really fun to do here, where you have this sheer drop of a light trap that I made in just one night of soak time. You can see all the cool little animals that pop up, including little squiggly worms that I hate, octopus. There's mantis shrimp post larvae in there, which are super cool. Lots of different filefish, a ton of snapper, and a few little French angels in there as well, which are my favorite post larvae. We didn't find any, but frogfish and an octo was cool. All right, so now let's move from the benthic habitat, so these are the reefs and everything, out into the open ocean, and talk a little bit more about larval dispersal. All right. So I mentioned earlier that lobster love to ride on jellyfish. Black water dives are cool. You can find all sorts of neat things, but there's nothing cooler than a phyllosome. And you can see here how flat they are. Here's another short video that hopefully goes now. Okay, that's talking about some of their behavior that they do. It's also written, so I can just talk over it. But look how small these animals are too. And that's just on a finger and super flat. And they're eating jellyfish, which is mostly water, in order to develop. It's super cool. They ride them like a little pony or something. Cool. So how do we study something that's super, super fragile, super, super small, and spends way too much time out in the open ocean? We can't. So instead, we turn to individual-based biophysical Lagrangian models of larval transport. which is a mouthful to say, but it's actually a really simple thing. It's a model. Models predict things. It's Lagrangian, which is a fancy word for movement, and it's biophysical. So we're able to put different features of the physical ocean, so we use US Navy models of the ocean, and seed it with digital larvae to track where they should go, depending on their behavior. Cool. We put a bunch of stuff in there. Don't need to go into it other than it's a global encompassing model. And then we include things like where the reefs are, when the animals are spawning, what they do in the ocean. Put it all together, and what comes out? Lots of little trajectories, right? So we release millions upon millions of little digital particles and create a probability of where things go. We know where we released it, we know where it ends up, so we can predict how these different areas in the ocean are linked. So Mark and I did this for Caribbean spiny lobster for about 5 years. And you can see this seasonal plume of larvae that gets pushed through this Caribbean current up and around, connecting the entire area. Cool. Why did we use lobsters, other than the fact that they're super, super cool? They're also worth a lot of money. And that means that you can get funded. All right, so what did we do? 5 years of larval transport with nearly 1 billion larvae and a couple of different scenarios. I'm just going to focus on one simple thing. Remember how earlier I told you about the larvae going up and down every day? What happens if you cut that behavior out? So we ran without and with that behavior. Red is more larvae in that area. And what we noticed is that the passive particles get ripped out of the system, versus the animals that have the behavior are actually able to swim underneath the powerful boundary currents and link the entire Caribbean, staying in the appropriate habitat while eating their jellyfish and getting happy and big. So there's a haiku about it. All right, so a model is just a model, unless you have some way of validating it. So we teamed up with some geneticists from the Smithsonian Institute, and we sampled a lobster at 17 areas throughout the Caribbean and looked to see how the predictions from the model linked up to the genetics, and we had high confidence. That was actually, normally this is a Bahamas talk, so don't worry about those. But yeah, we were able to predict and verify the fact that our model was working. We then used it to design where we think you should be managing these animals. So what are the boundaries for the fishery? What should they be? And basically the Caribbean sank out into 3 different basins. You have a northern area, a central area, and a far southwestern area. That the northern area receives all these different colors. This is just where each site, there's like 400 sites, received animals from, and the color is where that was from. So you see there's this consistent upstream effect. Then through the central, it acts as like this keystone, and then southwest coast of Central America is pretty self-reliant. Other people try to argue that perhaps national rules for managing lobster populations are the best. So we reanalyzed all of our data and reran a simulation with basically regional, in some cases, and generally national borders. And what we found is this crazy rainbow of chaos, which means that there's no way that you can manage the population based on your local stock, right? So here's just another way of looking at that. It's this huge, tangled, twisted web of chaos, as opposed to nice, orderly, ecological management. Neato. Another way of thinking about this is dividing the entire Caribbean islands up into sources and sinks. A source being somewhere that produces all of this awesome breeding effort and sends it successfully to other places. A sink being somewhere that is receiving a bunch of larvae from elsewhere, but its own animals are just going off and dying. So this is what it would look like in some theoretical metapopulation. But if we start messing with that, like humans are apt to do by putting boundaries on things that are arbitrary, then you can see that you can get some really twisted results, whereby one nation is entirely reliant on another for all of its animals, and its fishery, therefore, is dependent on resources outside of its own bounds. We did that throughout the whole Caribbean. And what we found is that some areas, such as Nicaragua, which is a huge source elsewhere, and Florida, which is a huge sink, are perhaps should be financially vested in each other. At the same time, if you look at areas such as where we are in Ceiba, the size of these bubbles is super small. That means that the local population is more or less tied to itself. Cool. So that means that local management, and for example, the MPA that is established here, likely has dividends. All right. Now, from lobster on to everything else. There's all sorts of cool little larvae here, but the key here is that these animals and this process really is the foundation of a marine ecosystem. If you lose the foundation, the whole thing crumbles. So now we're going to talk about something that is near and dear to my heart, probably the most charismatic piece of megafauna that you're ever going to see. The queen conch. Holy moly, what a cool thing. I mean, just look at those eyes. Yeah, and the awesome, intricate camouflage. But you know, it's not the outside that counts. It's what's on the inside. In this case, tasty, tasty conch flesh. And I work in the Bahamas, where this is a established part of the culture, and I'm going to let the top recording artists in the Bahamas tell you more.
5 seconds. 4, 3.
And the kids remember this because it's an earworm.
Conch, our national dish, has now become extinct throughout the Bahamas. Good evening, Bahamas. I'm Jiminy Swain. This is the Bahamas Tonight, the National Report. Restaurants are closing down and fishermen are going bankrupt. Environmentalists say that this is a catastrophe.
If this isn't caught in your head by the end of the night, something's wrong with you.
I've been diving, heard them say resources slow. I noticed things was getting slow, but man, I really thought that they was lying. How our delicacy could ever become mystery when all around me is the sea. My, oh my, should've left them babies in the sand.
Don't eat the young.
Why, oh, why? Should've give them chance to grow up, should've give them chance to multiply. We must think about tomorrow.
There's some simple rules to follow. Preservation, moderation. We need some conservation.
Come on. It's a bit that too long, even more people wanna come on. Let them go. If you know before you know, come on.
Mama is crying.
Built our lives up on the queen. Now not one piece can be seen. So how are we supposed to feed our children? A booming industry of shells and pearls and beauties of the sea. Now it's just a memory.
If we don't take heed, if we don't slow down, if we mind our greed, this will be our song. If we don't take heed.
So we'll turn around and all the corn gone. We must think about tomorrow. There's some simple rules to follow. Preservation, moderation. We need some conservation. Come on. It took me that too long, leave them there before all the come on. Let them make, let them grow. If you know before you know. Come on.
Chilling at the fry like hot.
And this was on the radio in the Bahamas for a while.
Crack up and fritters and plenty of sauce. And when I'm done with it, just send me some more. But you saying no conch though. But I cannot believe it, just cannot perceive it. They gotta be lying, look how big this is for real. I like boy with the deal.
It's about conservation, let the count them grow. Watch out for the poachers in the water slow. We use up so much you can count them low. Not import where we use to grow. Everything have a reason. Dive the conch in a season. No, them a face extinction. Can't even dive conch when we sleeping.
Preservation, moderation. We need some conservation.
And I love the commitment of the Bahamas. So how awesome is that? And the two main leads there, Dylan and Wendy, are super, super into protecting the local resources and the culture that goes along with it. So yeah, superstar group. They're telling me conch gone. I don't believe them. I'm a scientist. So I wanted to go out and see, are these conch populations really in trouble? You have to find conch. Here's a conch. Beautiful natural camouflage. All about those eyes, though. So how did we find them? We got dragged behind boats. A lot. I've gone hundreds of kilometers over shallow seagrass beds looking for conch at approximately tiger shark trawling speed in the largest shark sanctuary in the Caribbean. Here's what it looks like. So we take our research vessel. The shed has a 90-foot research vessel that moves in and out of the Bahamas, typically. And we have a whole group of 12 scientists that are hosted on it. If you're a dive professional and you ever want to come to the Bahamas and do this, holler at me. Basically, you get dragged behind a boat. Look for conch. You give you a golf clicker if you can't count to 100. You almost never find 100. Really easy, good on the back. Hold your breath and go. When you start feeling like you need air, go back on up to the surface and grab yourself a breath. The only real hazard are, of course, dolphins, which are way too friendly and get way too close. Yeah, anyone can do it. We've had probably 90 people out doing this. Cool. What did we find? So here on the bottom, I have fishing pressure. More fishing off towards the right. And then on the top, we have how many conch we were finding in an area represented as a density. Wouldn't you know it? More fishing, fewer conch. I'm an optimist, so I'm going to focus on the areas where these populations are remaining. These are areas that are super remote or in marine reserves, which is a solution towards a problem. First, just because we published a paper earlier this year that included Saba as well. There are large populations of conch on extremely remote banks. For example, the Keysal Bank, which is in between Cuba, Florida, and the Bahamas and owned by the Bahamas. We went there for 2 weeks, did a couple hundred insane dives, and found more conch than have ever been recorded in modern history. Eric, who is on Statia, Eric Bowman, I think he runs the national park there or something, has done a similar thing with gliders over the Saba Bank. Cool. Summary haiku for you. I like this one. Yeah. All right. Come on. So we have a problem, right? Tasty snails. Yum. They're disappearing. Oh, sad. Is the solution to protect part of the population and then rely on the magic of larval dispersal so that we could get a replenishment for the areas that are fished? The idea being that you protect the breeders and hopefully that will restore missing populations? Well, we can test that. The first thing you want to do when you're talking about a network of marine preserves is talk to the people. They matter more than anything else. You're not going to get a successful reserve if you don't have local community backing, so that's what we did. We had a bunch of community meetings. I really love this one. Don't wait until the cows come home. Yeah, come on. Yeah. That's a good one. But there's a really good mixed feel about parks throughout the Bahamas. So we wanted to go and provide a concrete example about how one of their marine protected areas is replenishing conch populations. So here's a rough example of what we're thinking. Say you got a fishing ground and a bunch of conch get taken. You have an MPA, and you want to know whether or not that's going to restore that fishing ground and that population. What are some of the things you need? First, you have to know how many conch are there. So, wouldn't you know it, you can get dragged behind a board, a boat, and count them. At the same time, you also want to measure these animals. So you want to know how big they are and how thick their shells are. You also want to know what their larval dispersal patterns are like. So is this marine protected area population self-seeding? Is it reliant upon itself? Or is there some mystery other population that could be providing the young into the park? So, in order to answer some of these questions, we went out and counted a bunch of conch, and then we also measured a bunch of conch. I love the little fishy coming at the GoPro. Oh, yeah. I think we've measured something like 12,000 conch over the past 7 years. It's a lot of conch. So we measure how big the animal is, and then we also measure how thick their shell is. All right? And you can see that was a pretty thin-lipped shell, and this one's much thicker. So that's really important data because the thickness of the shell lets us know how old the animal is. Think about it like rings in a tree. Only, unlike a tree, you don't have to hurt the conch. You just grab it for a second, take the measurement, put it back on its happy way, and then you have the data you need. Here's just another example of some of the thicknesses. So you can see here's a young baby spilling out of its shell adorably. And here's some rock-like adults, super old. Tremendous amount of variability. These are sea turtle and ray snacks. These are sea turtle snacks. And nurse sharks actually come up and suck them out. So the place that I decided to, speaking of nurse sharks, the place that I studied this was the Exuma Keys Land and Sea Park, which was kind enough to host me for a few months on a couple of years. And it's a beautiful place to work, but people have been also working there for years. So back when I was in kindergarten, people were measuring conch there. So there's this incredible database through the years that you can use to track to see how that population is changing. And here, the red line indicates the average sort of age of the population, and you see that it's advancing towards older and older ages with each successive survey. At the same time, we noticed that all of a sudden, the young adults were disappearing. What's going on? So we know populations are declining, and we know the age is increasing with year, right? So they're getting older. So basically, you have this geriatric population of queen conch. They're still great breeders, by the way. They know what's up. And we want to know why. We want to know why this population is turning into predominantly old adults. Where does the next one go? So we expanded our survey using scuba, which is way more friendly in the ripping currents of the Exumas. We work in buddy pairs, and we have a GPS on a dive flag and do a ton of drift dives that were really delightful. And we were answering questions about how abundance, size, and age shifted throughout an island chain with a marine protected area. Why did we use channels to do this? This is a natural choke point, so the babies settle out on shallow seagrass beds, and then as they grow a thicker and larger shell that prevents fish from slurping them up and sea turtles or other things from breaking into them, they start moving down into deeper waters with bigger predators, and there's lots of predators there. Also, this is a sort of natural barrier to fishing, because if you're free diving down for a conch and then your boat is way the heck over somewhere else, that's not so easy when you're carrying around a bunch of 7-pound shells. All right, so strong currents, and then also the locals tend to be scared of the big sharks and stuff that come through. Here's me measuring a conch. Oh, so elegant, so graceful, just amazing. And just like that, another data point is added. Cool. Here are 2 conch that are mating. Here's the male. This is how they attach. He's holding on, releases, and he's still holding on. So that's actually a huge part about why these animals need to be in a large density. So think about a slow-moving snail that has internal reproduction. If there's not a lot of them in one area, they're not going to bump into each other to breed. So as fishing reduces the population, breeding also decreases, sort of a cascading negative impact. So here's our study area. And come on, here's the park. And here are all the dives that we did. Just white equals dive, little red dots are juveniles that we found, and little black dots are adults. We did a lot of dives. We don't need to look at all of them, whatever. So let's get to the questions that we answered. So first one, are conch more abundant inside of the park? Here's the same data. Here split up by different channels. Right, so 25 different channels that we hit over a 90-kilometer span. Here, histograms will show up. This is north of the park, so those 7, inside the park, and then south of the park. And the captions here, no flare means young adult. Flare means likely adult. Breeding is self-explanatory. So we found young adults, or young conch throughout the range. Most of the adults were inside of the park, like a shocking change. And wouldn't you know it, where the adults are is where the breeding happens. So we can answer our question and say, yes, there's more in the park and they're breeding. Yay! How about that marine protected area? Does having an on-site warden going around with a machine gun help repel poachers? Probably. So we measured about 2,000 conch in this experiment. And the idea was that the closer you get to where the warden leaves every day, the more animals, the older animals, and the bigger animals you should have. And wouldn't you know it, that's exactly what we found. Cool. So a park works when it's well enforced, like here, where I couldn't touch things. So there's a summary haiku for you. All right, so we have our problem. We have tasty snails that are disappearing, and we have another problem now. Where is the next generation in the park? And could a possible solution be to protect more parts of the population in a strategic manner so that you have an interconnected network of marine parks that exchange larvae for an enduring and sustainable chain? So I modeled larval dispersal through the Exuma Sound. And here's the park. And it's a 2-gyre system. Basically, most of the larvae end up towards the north, and it's reliant on populations to the south that unfortunately are basically gone. And we verified this later. I'm just going to skip through this. Doesn't matter. Verified this later on another cruise where we did another couple hundred dives through the far north of the Exuma Sound, where we saw lots of whales, and then the far south. And wouldn't you know it, where we thought there was a sink, and where we thought there was a source population, or a hypothetical source, was completely fished out. There was almost no conch. And where the sink was, also where some kind gentlemen from Eleuthera go out on a sandbank with pickup truck and grab babies every day, there's lots of conch there. So that means that the park is fueling that fishery, and our model was working as predicted. Cool. All right, some conclusions for you, just to keep me on time here. The park worked. Yay, that was awesome. Marine parks work if they're actually well enforced. Again, enforcement has a positive impact on these animals. However, we saw that the larval sources into that park were to the south in areas that had been fished out. However, it's still working to fuel a fishery towards the north. And we saw our hypothesis answered later down the road. So what was really cool is we published a couple of papers on these results, and working for a nonprofit aquarium, every once in a while we get a little bit of media attention, and this story got picked up. And that forced the government of the Bahamas to react. So someone who actually had power to do something made a bunch of statements. And wouldn't you know it, the Fisheries Act of 2020 put all of these things, all of the recommendations that we made, into law. So there's my presentation as haiku. I'm at 42 minutes, so that's great. I have time for questions, which is great. But first, I really want to thank Claire so much for the incredible accommodations, if she's here, I don't know, and the hospitality. And also, the dive masters and captains, like the DJ team was incredible, finding everything, and it was a true delight. And all of you that I've met, thank you so much for being friendly people. So I have time for a couple of questions. So who has questions? I came all this way. Ask questions. Okay. That's Dan.
Hi, Andy. I'm a dive professional in the Bahamas.
Hi, Dan.
I'm showing the last data. You mentioned noise pollution being a problem. What is being done to help?
So that's a great question. It's basically what is being done to remediate the noise pollution in the ocean, which is impacting all these different life forms. So there's studies that have been shown that the anthropause, the Great Human Confinement Experiment of 2020, has decreased ocean noise, shipping noise, and led to positive impacts on things like dolphins that I don't really care about as much. Also, it put this cruise ship industry at a complete standstill. And in the Bahamas, I was still able to go and do my work, and they had a line of about 30 cruise ships anchored up on the seagrass beds where I wanted to work and wasn't allowed to, just sitting there with a bare bones crew. So in terms of regulations, I think there's a growing surge, just like for plastic, to make sure that there's something that can be done via especially shipping. But beyond that, who knows? It's an exciting thing for these kids to answer in the future, and all of us to be a little more respectful when we're out. I don't know, how do you make your diesel engine quieter? Go electric or use it less, more efficiently. Okay, next question.
Is there a science to part? Meaning, any part would be good, but I was fascinated by the dispersal. So you're saying that the part is working, but it's not creating more potential fisheries outside of that. So is there like a daisy chain or a leapfrog plan for creating parks along the dispersal path?
So creating a park, so the question is, how do you best create a park, and are there bad parks? So how do you optimize park placement? Well, it's an extremely complex thing, because if you look at, for example, the U.S. National Park system, it's all on the abandoned land that no one wants, right? So, okay, you want it, but that's not necessarily the best habitat for these animals. So we can make suggestions about where the optimal park would be, and something like the United States, NOAA has an incredible management plan that rotates, for example, different fish harvest in rivers in Alaska, depending on year and real-time counts and stuff. So there's ways that you can optimize harvest and to place a park properly. Sometimes it just takes time. So there were a few freshly minted parks that have decent protection that were on really poor quality habitat in the Bahamas that I surveyed the year that they were created. And I surveyed 5 years later and saw absolutely no difference. But it's a waiting game. And that makes it really hard too when you're promising all these grand effects and then they don't come through right away. It's really hard on the people who've made a sacrifice to make that park. So yeah, it's all about community and figuring out the best way of engaging the people who would in a place to benefit and their ecosystems.
Did you notice much of that?
So I was just in the Keys this year, and I had a horse conch the size of my arm that was looting a nursery, just eating all the babies, sucks them up from the bottom. I didn't have time to go into some of the predation studies that I've done. Hint, I don't like sea turtles for a reason.
The predation by...
A lot. So the average conch, after it's out of the larvae, is dead as well. It's about 99% annual mortality. Most of that is happening from crabs, octopus, lobsters, rays, turtles, and other mollusks. So murex and horse conch.
for that coast where, said that they were being attracted to by smell or whatever to go to the coast. From an evolutionary point of view, that's not very smart. That's where they have the most chance to be captured by fish. Is there any research that focuses and has tried to focus on changes in that sort of behavior? Maybe make more sense to go to the garden, something like that.
That's where we're going this year. Okay, this is going to be the last question, to repeat the question. It's basically, it seems really stupid from an evolutionary perspective to rely on an odor cue to get back to the coast, and coasts are dangerous. However, the open ocean is way more dangerous. You can't settle out there. So these animals need to also settle in and thrive in an area where there's plenty of prey for them. I don't remember the second part. Then is there research being done on whether or not climate is impacting these cues? And wouldn't you know it, if you use an extreme test and put different fish larvae under different ocean acidification conditions, it really messes with the ones that are not control. And it inhibits all sorts of abilities, from growth rates to odor to hearing. So yes, there's research being done, and it turns out that rapid changes are bad. Pollution is also a huge thing, not just plastic, but a lot of, in Florida especially, runoff creates different sex ratios and wreaks havoc with a lot of different animals. Okay. One more question? Question? Come on, cool away here. I want some questions. Hi. Yeah, so the question is, does this sort of model work for other species, such as locally diadema? Yeah, it has been done. So there's the classic study by Lesios is basically how they mapped out the currents. I've run everything from, part of my PhD was funded by the oil companies, bless their hearts, for looking at oil remediation. So if there's an oil spill here, where is it going to go? What ecosystem is going to impact? So we've done everything from 3 days to 2 years worth of dispersal. So chances are, yes, it could be done. It's not catastrophically hard to be done if there's a little bit of money. Also, locally, you could... What? Locally, though, you could throw drifters or something in just to see what's happening on a short scale from where you expect the high urchin populations to be, to see whether they're entrained behind the island or not. That would be cool.
All right, guys. Huge thank you to Andy for coming out and giving that awesome presentation. This has been a fantastic See and Learn this month, but we are not done just yet. Now, we do have the raffle left. I'm sure a lot of you guys here are in it, so make some noise if you're in the raffle tonight. Right? Awesome, guys. So we are going to start. Emily is going to come up and pull the tickets. All right. So if any of you do not like that you didn't win, please blame Emily. All right. Do not blame me. Okay. So we're going to start to go through some of the awesome prizes that we do have here. Oh, she's putting the blame on someone else. I see. On the kids. Blame the children. Got it. Now, we've got some awesome prizes this year. Again, I already went over them, but to start out the night, we have a signed copy of Dr. Edie Witter's brand new book, Below the Edge of Darkness. So, Emily, who is the winner? Drum roll. It's pretty weak drum roll for Daniel from SCS. Here, big round of applause. May not be here right now. Sounds like a school teacher, so working pretty hard. you can win even if you're not here or if you're viewing online from home, all right? Now that we've started drawing tickets, it is too late to buy any, but we do still accept bribes, all right? So please let me know, or my Venmo is... I'm kidding. All right, so Daniel, if you are watching, wherever you are, get in contact with Lynn and Emily, and we'll get you your signed copy of the book. All right, now the second prize that we have tonight is a goodie basket of some natural things that are made on Saba. So, who is the lucky recipient? It is Brian Vargo. Here? Brian Vargo, wherever you are, get in contact with Lynn and Emily. All right. Now, the next prize that we have tonight is a class with Joe Bean. I believe she just got back on the island, so I'm sure she's ready to get back to some glass making. Now, who's been pulling these tickets? Is it you guys? Okay, awesome. So you know they're good. It hasn't been Emily, don't worry. All right, and this one is Erica Moulton, who was one of our speakers earlier in the month. So Erica Moulton, she did the ROVs, put in a lot of good work. She gets a homemade class for next time she comes back to Saba. So that is awesome. Now, let me see the next prize. Is this right, Emily? Says a date with JJ. I think it's the next prize. I guess we got to do it. I'm kidding. I'm kidding. I'm worth much more than a $5 raffle ticket. Okay. The next real prize is one of our big ones, a 3-night stay at the Spyglass Villa. All right, one of the cottages that's here. So, big drum roll for this one. And the winner is Rory O'Connors. Woo! Yeah! Not here, maybe not here. I think I got that right. If I butcher your name, I am sorry. All right, but you won a 3-night stay at the cottage. So get in contact with Lynn and Emily. All right, the next one, which we have here tonight, is a handmade indigo pareo from Anna Keene. All right. It's a gorgeous piece of art, so we'll be giving that away. Who is the next winner? And the next winner is Cloudbreak. Is that the hotel? Casey and Steve. Maybe they can hang it up in Cloudbreak because they wrote the ticket. I don't know. That'd be pretty cool. So awesome. A guided hike from SCF. We got some SCF people in the house right now. All right. So let's see who wins this guided hike. And the winner is Frank? Phone number. I can't even read the phone number. 491-7144. German Frank. Who is German Frank? Woo! All right, awesome. Now, the very next prize is another one of our 3-night stays at a cottage. All right, so Emily, who's going to pick this ticket? What? I don't know. This is up to you. It's one of the owner of Long Haul. Why don't you come up and pick this ticket? You've been giving us a lot of sponsorship this time. Let's see. Woo! And it is Mike Butler, who is one of our... What? Mark Butler! Cut that out of the live video. He was one of our speakers earlier, actually worked with Andy and recommended Andy to us to come here, so well deserved. He's going to be back on Saba to claim his 3 nights, so you guys can talk to him more then. All right, now we have some gorgeous handmade jewelry, and it's all made by Marie, who is here. Give a big, come on. She can pick it. Who gets your jewelry tonight? Emily's laughing, and it doesn't make me feel good. The winner is the GNT genie himself, Duco! He is probably hard at work over at Shea Booba right now, but he'll get some lovely jewelry that he can wear around town. You know, nothing wrong with that. So, the next prize, another 3-night stay at the Compass Cottage. So, big drum roll for this one. Emily wants to pick this one. Do you guys trust Emily? Yeah. Some yeses, I heard some no's. And the winner is Gisela from SCS, one of the teachers. Woo! All right. I'm sure that'll be very necessary after dealing with you hooligans all day, right? Now, this is another one of our big ones. We have John right over here. Big wave. Nope, he's hiding. He is going to make a custom hand-forged knife for the winner. John, do you want to pick this one for us? Who is it, John? John. It is Mike Holter? Mike Holter! Woo! Awesome! That is incredible. I think the first person that's here tonight that's won, right? That is awesome. Aren't you glad you hosted us tonight? I told you I'd take bribes. This is what it took, okay? And for a 3-night stay with our very own Larry and Sherry, the owners of Flamboyant Cottage. Big round of applause for those guys for giving us such a great prize. So, do you guys want to jointly pick the ticket? You both have to decide on the same one. Or she has to approve it. We'll see. Oh. All right, and the winner is Kim Hull, who is one of our presenters from earlier this month. So she will be back on Saba. She talked about some spotted eagle rays. Her associate teen talked about some small-eyed stingrays. Some pretty cool stuff. So they will be back on Saba very, very soon. Now, for something that a lot of people have put in some money for, right? Who here tonight is hoping to win this? See, we've got some people praying right now. I guess that works. I don't really know. All right, an 8-day, 7-night adventure on the Caribbean Explorer. Okay, so, Emily, who's going to pick this one? Do I? Oh, I'd be honored. Oh. Digging from the very bottom. Ready? Big drum roll for this one. Ready? And the winner is Dr. L. Woo! Yeah! Right in the front row. That is awesome. Maybe you'll take your kids out with you or something like that. But that is our raffle prizes for this year. We'll have a bunch more next year, so please come out. And thank you guys so much for doing See and Learn with us this year. All right. Now, again, we do have to turn this place back into a restaurant, so please head to the bar, head out into the street, don't get hit, and join us for dinner tonight as we thank Long Haul. And a big thank you to Long Haul real quick. Big round of applause. Thank you for sponsoring us.